Namaste students, I am JVN Dr. Aziz Mahmood Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic mechanical method of the cell disruption. So in the previous video I discussed about what is the cell disruption and what are the basic types, what are the basic methods of the cell disruption. So there are some mechanical and non-mechanical methods which are used in the cell disruption. The basic principle of each method will be discussed in next videos. And in this video, I am going to discuss about the mechanical method of the cell disruption. The topic describe what are the mechanical methods which are used. So first, what is the principle of mechanical methods? In the mechanical method, cells are subjected to high stress via pressure, abrasion, and with rapid agitation with beads or some ultrasound methods. So these are the mechanical methods. And the method of disruption include the cavitation, shearing, imprisonment, and the combination of these methods. So mechanical shearing and some this will cause applying pressure, abrasion, agitation will lead the cells to die and they will be disrupted. Their cellular components will be come out and the cell will be disrupted. Similarly, intense cooling of the suspense after the treatment is required in order to remove the heat generated by the dissipation of the mechanical energy. Some high pressure methods can be applied in laboratory, such as the French press and the Hugh press. So these methods can be applied. First method of mechanical separation is the bead mills. Okay, bead bead as the word indicates, there are some beads and the cells are mixed with some beads. In this method, a jacketed grinding chamber is used with a rotating shaft, and with rotating shaft is inside. It is between the chamber at the center of the chamber. So agitators are fitted in the shaft. So there is a shaft, and in the shaft there are some agitators. These agitators are at the center and provide kinetic energy to small beads, and that are present in the chamber, which makes the beads collide with each other. So when the agitator will get rotated, then the beads will collide with each other and they will come to each other. When the glass Bedoni and stainless steel balls are used in this method, the size range being selected for most effective release of the enzyme. The increased number of beads increase the degree of the disruption. When we have added more number of the beads with cells, then they will lead to the more disruption of the cells. And the low number of the beads will be will result in the low disruption of the cells. Similarly, some heating and power has also effect on the cell disruption. If we are using more power, then the cell will be disrupted. If we are giving more heat, then the cell will be disrupted. and at the end the cell will get synced, and after that the cellular compartments will come out. The variable factors which affect the cell reception by the glass beads include the agitator speed, the proportion of beads, that how much the beads are there. They are small, they are in low quantity or high quantity, the size of the beads, and the cell suspension concentration, the cell suspension flow, and the agitator disk design. So these are the factors which affect the cell suspension and debris, cell disruption. Some drawback are also there in this method of glass beads this glass beads cell disruption. The first is the high temperature uh, can increase bead volume, and so additional cooling systems are required. So if additional cooling system is required, then it may increase the cost. Another disadvantage, another drawback is they have poor scale up. This method has poor scale up. And similarly, the third thing, which is the contamination rate is high. Chances of the contamination is very high. Now, the next technique, which is the ultrasound. It is also a mechanical method. Ultrasound disruption is caused by the ultrasonic vibrators that produce a high frequency sound. Okay, when the high frequency sound is there, then a wave density of about 20 kilohertz is flown, which results in the cell disruption. A transducer then converts the waves into the mechanical oscillations through a titanium probe, which emits into the cell suspension. Then they are very effective in small scale work, and they will cause the cell to shrink. First, what is the mechanism of the cell disruption? When we talk about the mechanism of the cell disruption, first the cell is like round shape. Okay, cell is like this round shape. After that, the cell will get shrink here, like okay here, and then the cellular portion, this portion will be another shrink, then they will shrink, and at the end the cell will go for the lysis in this manner. So the two cells will be created in this manner, and all the materials will be, all the cells will be divided. There will be jetting in the cells, which will allow the cells to divide, and all the cellular compartments will be released. What are the drawbacks of this method? The upscaling is very poor. This is used for the small scale. It has higher energy requirement. Okay, high health and safety issues are there with this method, because they will cause very high noise, and it may also cause the person which are going to perform this experiment, it will harm them, and this experiment is not continuous. The next is the French pass and the high pressure homogenizer. In a French press and high pressure homogenization, and the cell suspension is thrown through a valve into a pump cylinder, and then it is forced under pressure of 1500 bar. So in narrow annular pump, and the discharge valve where the pressure drops the atmosphere. The cell disruption is achieved by the sudden drop in the pressure upon the discharge, causing the cell to explode. So in the French press method, a homogenizer is used which will allow the cells to explode at the end. Operating the press at high pressure, the number of passes in the slurry through it can be decreased in order to obtain the desired degree of the disruption, and the operating pressure may be limited due to the deactivation of the certain heat sensitive proteins, which may increase the number of the passes. The factors which influence this method are such as the temperature, the intracellular location of the enzymes, the number of the passages, the operating pressure, and the biomass concentration. So these are the factors which affect the uh, functioning and the uh, cell disruption. Now, how high pressure homogenizers. This system consists of the displacement pump which throws the cell suspension through a check valve into the pump cylinder. A high pressure of up to 150 millipascal and flow rate of the 10,000 liter per hour is applied. The main disruptive factor is pressure applied and pressure drop across the valve. The higher the operating pressure, the more efficient the cell will be found. Multiple press decreases the throughput productivity rate and they result in the fine debris which is difficult to remove further downstream. It is used at high pressure compatible with relatively safety of equipment and temperature stability of the enzyme. The valve unit is prone to erosion and must be well maintained. So I explained here some mechanical methods. So what kind of questions which can be asked here? The examiner can ask you explain the mechanical method of the cell disruption, explain the techniques which are used in the mechanical cell disruption methods. They can also ask about the drawback of these methods, such as uh, glass beads, homogenization, and they can also ask about ultrasound, explain French press, high pressure homogenization, explain high pressure homogenization. So these questions can be asked. I hope and students can better answer all these questions. If you have any questions, you can ask me. Thank you very much.